The basic model for the DNA structure was developed by Watson and Crick. As seen with the clay DNA model, DNA is held together with a negatively charged sugar phosphate backbone, brown being the sugar and black being the phosphate, thus causing DNA to be negatively charged. Each strand of DNA is anti-parallel to the other strand, as the 5' to 3' strand is associated with the 3' to 5' strand. This can be seen similar to a two-lane street as cars pass by. The sugar component is what binds to the nitrogenous bases. These bases include adenine in orange and guanine in red, which are purines, as well as cytosine in green and thymine in blue, which are pyrimidines. The basic structures for purines and pyrimidines are shown below. Purines consist of two cyclochains, whereas pyrimidines are a smaller molecule as they consist of one cyclic chain. Watson and Crick noted that adenine pairs with thymine and guanine pairs with cytosine, and in between each pairing, the bases are bonded together with hydrogen bonds. These pairs are also stacked upon each other within the helical matrix. Bases are hydrophobic, meaning they hate water. So in order to reduce the amount of water exposure to the bases, they stack on top of each other tightly as if you were to wring out a wet towel soaked in water. DNA's helical shape also gives it rise to regions known as grooves, or major and minor grooves. These grooves are how different proteins interact with the bases. Proteins much prefer the major grooves as a much larger space to interact in compared to the minor grooves. The deoxyribose and ribose sugars are composed of 5 carbons, 4 of them being of significance. Carbon 1 is what binds to our bases. Carbon 2 is what determines whether the sugar is in RNA or DNA. If carbon 2 is bonded to 2 hydrogens, then it is in DNA. If it is bonded to an OH group, then it is in RNA. Carbon 3 is what will bind to carbon 5 as polymers are made. Lastly, carbon 5 is what binds to our phosphate group. So let's say DNA is placed into a saline sodium citrate. The salt ions can partially neutralize the negatively charged phosphate backbone by interacting with the negative oxygen on the phosphate group. As DNA neutralizes, this allows the Tm to increase. Tm is when half of the molecule is denatured due to heat. Because of the salt buffer, DNA would denature at a much higher temperature than normal.